let's go with the next step, which is prediction of physiochemical properties of antigenic protein. Okay, so that's what our next step is. So now we have finished with the functional properties. Okay, we have reached till step 3. Next is prediction of physiochemical properties of antigenic protein. Okay, that's what our step 4 is, to predict the physiochemical properties of it. So here also let's go with the 2 servers. First go to prot-param tool. After going to prot-param tool, you just have to paste the sequence, delete the FASTA header and click compute parameters. So let's go to this particular tool and proceed with the other steps. So either go to the link or check for prot-param tool and click on the first result, which says prot-param tool XPESI. So this is our site. So let's enter. So it's a tool which allows computation of various physical and chemical parameters, right? So it says please note that you may only fill out one of the following fields at a time. Yeah. So if you want, you can enter the accession number or sequence identifier like this, but we are not giving anything. Either you can do that, or you can paste your own amino acid sequence in 1-letter code in the box below, which means in 1-letter code, which means you need to remove the FASTA header. Okay, so either you take only the protein sequence, the plain text format, or copy it and delete the FASTA header separately. And then click for compute parameters. Then you will be getting the result. So in the results, you can see the sequence. Here the sequence is mentioned. Then you can see the number of amino acids, which is 208. You have 208 amino acids in your sequence. Then the molecular weight of your protein, which is 22,950.08. Okay, that's your molecular weight of your protein. Then you have theoretical pi, which means at its isoelectric point, or it means that the protein has no net charge because the positive and negative charges are equal. So at this point, that is 5.77, the protein has no net charge because both the positive and negative charges becomes equal. So the theoretical pi is based on the primary sequence, but it is not likely to match the actual pi because some of the charged side chains may be buried inside, or it may be present in the salt bridges. Okay, so theoretical pi is mentioned 5.77. Next, you can see the amino acid composition that is being written here. Okay, so if you want, you can take, or you can, if you click onto it, you will get it in CSV format. And the next is total number of positively and negatively charged residues are mentioned. Okay, 14, and there are 13 positively charged residues. Next, atomic composition is mentioned, its formula is mentioned, total number of atoms are mentioned. Then it also says about extension coefficient. What does it mean? It's a measurement of how strongly a molecular species absorbs light at a given wavelength. Okay, how strongly this particular species absorbs light. Here it's at 208 nanometer. Next, the absorbance value is also mentioned. When cysteine residues are formed and when they are not formed, that is also mentioned. Now, next is half-life. That is the time needed for the protein to disappear in the cell. Okay, that is also mentioned. The estimated half-life is also mentioned, which is 100 hours in this case, or in this protein. Now, the stability of your protein is also shown, okay, which says an instability index. That's what says about the stability of your protein. So if it's more than 40, then it is considered unstable. So the instability index is computed to be 41.65, which means it's more than 40. So it means that this particular protein is unstable. Okay. Now the next mentioning property is how much amount is covered by the aliphatic side chain. That is mentioned. Here it's 82.88. So by aliphatic side chain, it's about 82.88 that has been covered. Okay. Now, how much hydrophobic or hydrophilic it is, even that is mentioned in terms of grand average of hydropathicity. So, okay, so the value is minus 0.038. So if it was positive, it means it is hydrophilic. And if it is negative, it means it's hydrophobic. So it's minus 0.038, which means it's hydrophobic. Okay, so these are the values that has to be put in the Excel sheet. So in the Excel sheet, tabulate the number of amino acids, its molecular weight, theoretical pi, extension coefficient, which means when cysteine is present, when cysteine is absent, estimated half-life of it, of the mammalian reticulocytes here in this case, then instability index, instability nature, whether if it is stable or unstable, aliphatic index, and Gravy score. So ideally, anyway, since I'm just showing a demo, I'll be proceeding with this particular protein. But here in this server, it says unstable, right? So if it's unstable, please do not go with this protein. The protein which is stable has to be considered, okay? So when you do your exercise by checking the other different proteins, see if it is stable, then go for it, okay? Okay. So now let's plot all these values in the Excel sheet. So here I have mentioned all the headings: amino acid, molecular weight, pi, EC, T-half, instability, aliphatic index, and Gravy score. So how much was the amino acid number? It was 208. So copy and paste it here. 208. The molecular weight was 22950.08. Theoretical pi was 5.77. 5.77. Extension coefficient when the cysteine is present, it was it was 29380. Okay. Now when the cysteine residues are absent, it was 28880. Now the T-half was for mammalian reticulocytes in vitro. It was, or it is 100 hours. The instability index, when you look at it, the instability index is 41.65. It says it is unstable. So the nature is, it's unstable. Unstable. Then you have your aliphatic index, which is 82.88. So copy and paste it here. And the Gravy score, which says about the hydrophilicity and hydrophobicity, it's minus 0.038. So here, which says it's hydrophobic in nature. Okay, so these are the values of physiochemical properties of this particular antigenic protein. So now we have mentioned all the values. So do the same thing for the other proteins and write it down here. Okay. So this, we are making it an Excel sheet just to make sure which, or we are actually shortlisting the protein, right? So this is how you need to do. Okay. After plotting all these values, then you can have a look at all these proteins and then select which one is better. Okay. So that's our intention, to make everything plot in an Excel sheet, right? So this was about one software. Let's go to another server, okay, which is AA Prop. Let me just show you the slide. Okay. So we are going to this particular server, go to this particular link, biogem.org, and go to tools, AA Prop. So I have searched as AA prop tool. Let's go to the first result, which is protein physiochemical properties prediction tool. Again, it says enter amino acid sequence with 1 letter code. So this I've just tried with example protein sequence. Paste your plain protein sequence without the FASTA header and then click predict option. So here again the parameters considered are same as that of the previous one. You have the user provided sequence and the other informations. Okay, so just have to compare it. It's 208, same thing. It's 22950, same. Then the PI value is 5.77. Everything, the amino acid composition, all the list is mentioned here. But you have an addition. Okay, one addition is aromaticity. Let me just scroll down and show you. Yeah, you have aromaticity, which is mentioned. There was just aliphatic index that was mentioned, right? So this was the amount covered by the aliphatic side chain. Now aromatic, okay, how much of the aromatic chains? It's 11.5%. That is mentioned. And Kite and Doolittle hydrophobicity plot is also mentioned, which is an algorithm created to find out the hydrophilicity and hydrophobicity of the protein. Okay, the x-axis represents the position and the y-axis represents the score. So here this is the minus indicates the hydrophobic nature and the positive sign. So it starts from minus 2.8 to 1.956. So this is just a graphical representation. As you can see, score is mentioned. Position, since it's 208, you have 208 sequences till 208, and the window size, which is 9. Okay, that's the additional information. So in this lecture, what we studied, let me just scroll over and show you, is primary protein structure prediction, which is antigenic protein sequence retrieval, then functional properties of it. We have predicted the antigenicity of the protein and the allergic nature of the protein, and then prediction of the physiochemical properties. Okay, we know about the protein's antigenicity, allergenicity, and the physiochemical properties, and also the sequence of it, of course. Okay, so that's all about this session. Thank you.